DNA and protein to protein in detail. So last video we talked about the bone marrow and how inside the bone marrow early red blood cell, and inside that early red blood cell we have DNA. So let's magnify that DNA again. And remember, inside that DNA we have a code of different letters, A, T, G, and C. So now what's special or different about this particular process of making protein is we have this thing that comes in and reads the DNA in sections. This thing that just came in, we're going to call this RNA polymerase. RNA polymerase is the main guy that will translate this DNA code and make another code. This code that it makes is going to be called mRNA. M stands for messenger. RNA is a little bit different than DNA. In a sense, we're not going to talk about that difference, but it does still have a code that it makes. And I'm just going to write in these letters. They'll make more sense in a bit. So with DNA to RNA, it gets a little bit trickier. So I'll write out the pairings, DNA to RNA pairings. And the only slight difference is that A in DNA is going to pair to a U in DNA, or RNA. T is going to pair with A's, and G's are going to be with C's still, and C's are with G's. So just so we're clear, this section right here is DNA. This section right here represents RNA. So this code that is made, or this messenger RNA, has to go to another part of the cell. So that's where I'm going to draw the next thing. So this code is going through kind of a belt. We still have the code A, U, G, U, C, but instead now we have this thing that reads it and translates this code into something else. This thing is called a ribosome. Translates it, and it makes finally the protein. But still, we are not done yet. This protein has to be folded properly. And it has to be folded, like I showed you, into that proper shape of that hemoglobin. So this is good hemoglobin. However, if there's something wrong in this code, it still gets folded, but it gets folded into something we don't want, or it doesn't work. And we're going to call this bad hemoglobin. So bad hemoglobin actually forms these sickly cells inside the DNA, and this is sickle cell anemia, sickle cell red blood cell. Whereas this hemoglobin over here, this makes a proper hemoglobin shape, and therefore a healthy, normal red blood cell.